Well, can I say, first of all, what a pleasure it is to be back in Oxford as a Scotsman who was here for 5 years, living, doing my house jobs, and then a PhD, or DPhil, here at Oxford, and has been exiled in California, in the sunshine, for the last 12 years. It is really wonderful to be back here, and the weather was very Californian, as was spotted yesterday. This also gives me a chance to correct the spelling of personalized, which is something I know will be important to this audience. Apologies to Tal, but given we're in the homeland, we should do this right. Here's my disclosures. There'll be a little bit of more detail coming up about the company we started up a few years ago, Personalis, and as you can see from the names on there, no surprise that we're located in Silicon Valley in California. Well, one of the things I'm here today to tell you is that genomic medicine is here, at least in the part of the world that I live in, I think in the United States. It's here for rare disease because it is now no longer the future, it's no longer something theoretical, it's exciting, but it's actually here. And it's not just stories like the ones you might have seen in these articles here, but it's actually a reality in terms of everyday life. In clinic, we're sending exome and genome sequencing on individuals in the clinic, really on a daily basis from the healthcare system. And the question is, where do we go from there? But these stories, I think, are particularly interesting because they highlight kind of poster children, if you like. And I wanted to give you, we all sort of carry around one or two of these cases in our pockets just to pull out. And so I wanted to just give you one of those, since this is about medicine. Here's a little baby who was transferred into the Stanford Children's Hospital at 36 weeks of gestation because of suspected heart block. When the baby was born, we saw this ECG, and in fact, this was not heart block, but rather long QT syndrome with a corrected QTc in the high 500s. It led, of course, to the challenge of torsade de pointes, which you see in the bottom left of the screen, and multiple cardiac arrests on day 1 of life, followed by implantation of a defibrillator, shown here on the bottom right, and a bilateral stellate ganglionectomy. Essentially, what we do with these little kids is throw everything at them. But we know here that this is not a mystery diagnosis. The diagnosis here is known. It's long QT syndrome. But long QT is one of those, for those of you who don't know, is one of those diseases that is defined by its genetics. So we now know the genetic basis of the disease, and we send a genetic test. And the most common types are labeled 1, 2, and 3. 1 and 2 are potassium channel mutations. Long QT3 is a sodium channel mutation, and we have drugs that are specific for sodium channels, and more coming on. So it's an opportunity to be precise about our therapy. Now, in the current system, the technology has moved so fast that I can send a panel. It'll take about 2 to 3 months. It'll cost about $5,000, about £3,000. And I'll get a result. They'll sequence about 5 genes, mostly by Sanger sequencing. But in this case, we thought we could do better for a little baby that was really in extremis in the neonatal intensive care unit. So we took advantage of Illumina's fast track whole genome sequencing, which was completed in Tina Hamburg's lab in San Diego over the course of a weekend. And this returned the result shown here, which is a known variant in the KCNH2 gene, and another potential modifier in RNF207 that likely explained the baby's long QT. So we were able to deliver this result back in the course of 3 days, which is significantly better because also, as you've heard, this kind of genome sequencing can now be done, at least for the data cost of around $1,000. The clinical product is a little bit more than that. So we're really talking about 2 worlds that simultaneously are existing. One where we send the old style panels and it takes 3 months and costs £3,000, and one where it costs half of that and we get the whole genome. So it's a pretty incredible thing. And the end of the story is that she was doing, little Jaslyn shown here, is doing very well 6 to 8 months out now, and is fortunately on stable therapy. Well, this rapid turnaround whole genome sequencing is possible. It's actually a little faster than this now. Stephen Kingsmore at Kansas has worked closely with the folks at Illumina, and I think they have this library prep through a few tricks down to 18 hours in total to get to the data. We use RTG, real-time genomics, based in New Zealand for alignment and mapping. They're also about half that time now. And then the variant calling is sort of local Stanford tools. So it can be done pretty fast and is a reality in terms of things we could do. Well, that's a question for individuals with rare disease or with genetic disease where we can turn things around fast. A few years ago, we had this vision of when we sequenced the genome of one of my colleagues, Stephen Quake, and thought about interpreting his genome for medical use. Our use case at that point was what do we do when there's a genome in the medical record of every patient? We need better, other than panic, we need better tools, and we need to think about what we're going to do with that. And the question is, 5 years on from when we did that study, Are we there yet? And I think that we're not quite there yet. And I'd like to go over some of the challenges that might help us get from here to there over the course of the next few minutes. I think it's probably more likely going to be something like this from here to there, because I don't think it's going to be a straight line. Anyway, some of the challenges. This is Tommy Caldwell, this is Yosemite Park in California, and they recently climbed this dawn wall, which is, I was sweating actually just watching the YouTube video of him doing that, so if you're interested, the rock climbers among you. I'm not sure about sleeping in the tent shown here on the wall. Well, the first thing to remember is the genome is complex. I think we often forget that. It's in reality, because we have these fancy tools and we have lots of short reads that spit out and we align them to the genome, we sort of forget just how complex it is and how much of the genome is actually repeats. About 50% of the genome is repeats. The introns are about 40%. Retroviruses probably make up 8% of the genome. If we look at the non-unique, when we're back in the 35 base pair area, you can see there's a large portion, 15% of the genome, is non-uniquely mapped to 35 base pairs. This, of course, improves if you have longer reads. 100 base pairs, it's like 10%, but that's still a remarkably large number. This is after repeat masking. So I kind of get cold sweats when I think about this kind of thing. With pseudogenes and segmental duplications and paralogous sequence, we take short reads and then we think we know where they come from, but really we don't know where they come from a lot of the time, and there's no real consensus as to how to handle them. This work just published in Nature from Evan Eichler, looking at the hydatidiform mole using PAC biosequencing, gives us another look into what long reads might give us a sense of, and in red and black you can see here the large CNV events that have been missed for the most part by the short read sequencing events. So the new microscopes will give us a lot more detail, and I think we've heard some very interesting presentations even today on some of that new world ahead. Well, covering the genome is also consistently as hard, and we've lived in a world of exome sequencing up to now. This is from the EXACT server, which Dan MacArthur and colleagues even in the audience have been contributing to. So information on 64,000 exomes, the off-the-shelf exome capture kits. This is the KCNH2 gene. This is the one I just showed you that was responsible for long QT in the little baby. Well, this exon you'll see towards the end is not very well covered, which wouldn't be a problem except that there are 19 pathogenic variants known in that exome. So when you get an exome, you don't get an exome, you don't get a nice smooth line. You actually get a lot of holes. You get a lot of valleys and a lot of peaks. And this is a really major problem, because the data that you get in is the best. Your algorithm can only do as well as the data it's given. And so if you're not covering an area of the genome, you're never going to be able to cover, to report out a variant in that region, no matter how good your algorithm is. And so we did this study just last year on a small number of individuals, but comparing the, at the time, complete genomics platform with the Illumina platform and whole genome sequencing for the 56 genes that the American College of Medical Genetics had sort of deemed some of the most important, the ones they thought should be read out as incidental or secondary search findings. And if we were back in the days of Sanger, before a clinical report was read out, every single base pair, coding base pair, would be called. So this graph would look like a straight line down the left. This is number of percentage of the gene not called at a particular level, which in this case is 10-fold and a quality base pair quality of Q30. For those of you who follow that, and then the 56 genes, this would be a straight line down the left. So anytime this graph is towards the right of the y-axis, we are not calling a gene at clinical grade quality. And that's something that we really are focused on quite a lot, both in my lab at Stanford and in Personalis, the company that we started. So certain classes of variation are hard to call. We've lived in the GWAS era where we think about single nucleotide variants a lot. We're pretty good at those. You can see up here, 99% concordance between platforms. But insertions and deletions, which are, if anything, more likely to cause disease because they disrupt the genome more, 55% concordance. And if you narrow that to Mendelian disease, it's actually even lower. So we're not very good at calling indels. Also, rare and novel variation is normal. The large-scale sequencing studies have told us that, and it will be the case for some time. So we need to work out what we do with rare and novel variation. We're not going to be able to just look up a database and it'll have the answer because we've seen it before. This is from 3,000 individuals with hypertrophic cardiomyopathy, a Mendelian cardiac disease. And if you look at the number of people with a specific variant, you see some of the founder variants here, this 50 individuals. But there's not very few people with variants that have been seen many times. The vast majority of people have a variant that has never been seen before. And that's a lesson for us, because I think that we're going to need to make our algorithms as good as they can be in terms of prediction. And then clinical grade, if we're thinking about clinical grade, we need consistency in reporting. And this has been a challenge because we've come from an era where variants have been reported as pathogenic simply because they were variants in a gene that is known to cause a disease. When we did this study, first of all, we worked out that it took an average of about 54 minutes to do a manual annotation of one gene, one variant. But also when we had a subset overread by 5 individuals, we found that there was only moderate classification agreement between them. 70% of the genetic variants catalogued as disease-causing were reclassified, and in most cases to lesser significance. So those are the questions. That's the problem. You know, this is a kind of good news, bad news thing. So that was the bad news. What's the answer if we want to get to truly clinical grade sequencing? Well, there's a few things. First of all, we should define community standards, and publications like these that I contribute to with a group of really excellent folks thinking about what causality really means in the context of Mendelian disease. The American College of Medical Genetics has these guidelines. I'm sure there are equivalent guidelines here in Europe and the UK. So that's the first thing. The National Institute of Standards and Technology in the US, again, there are likely similar and equivalent ones here, have put together a set of gold standard variants that they nicely called genome in a bottle that many of us have used in order to use as a sort of gold standard set. But I'll show you in a minute, it's more of a bronze standard than a gold standard. But if you look at those 56 genes and look at their high confidence regions, again, you'd expect that, or you'd hope that all of those genes would be in high confidence regions. And as you can see from this graph, then large portions of these genes are in the low confidence regions. So we have quite a bit of work to do in terms of pushing forward. In fact, only 81% have shown here of the known pathogenic variants in ClinVar are in the high confidence regions. Well, I also would recommend sequencing families. I think you've seen great examples of that. Peter Donnelly talked about this earlier in terms of the UK project. A few years ago, we did this study, where we looked at and built some models, inheritance state models, on whole genome sequencing for this family. This is actually John West, who's actually the founder of Personalis, the startup company we have. And this takes vetting your CEO to a new level, because we sequenced, or he had originally sequenced his whole family and came to us with the idea of interpreting his family's genomes. And a lot of the work that was published in this paper became really the foundation of the company. But it's now used on a daily basis for families like this in our clinic at Stanford. And this is a family that had neurodevelopmental disorder and 2 brothers shown here. And one of the singletons, one of the brothers, had already had an exome sent to a very reputable lab, and no answer was found. When they came to our group, though, we thought that we would get more power from doing this family-based sequencing, and we did, and managed to narrow the search base significantly and find a gene that we were very excited about. We were getting ready to write up a new syndrome when, of course, the paper was published from another group showing that SNX14 in this case causes a distinctive cerebellar ataxia syndrome. So that was good because it saved us the work of actually going and doing all the basic science that would be required to prove this was the case. But also, I think, a learning point that sharing of phenotypes is just as important as sharing of genotypes. Shows the strength of family analysis. Here was the singleton. This was the case that the clinical lab who were trying to solve this case using the singleton. They had 450 genes to look at and 1,500 variants. Using the compound heterozygosity inheritance state model, using the trio, we could bring this down to 50 genes and maybe 30, maybe something like 100 variants. But using the quad, we could get it down to really a small number of genes, 10 or 15, and only about 20 variants, which, given an hour of manual curation per variant, is quite helpful. So what about that coverage problem? Well, this is something that I mentioned Personalis have been focused a lot on over the last 2 years. The standard in blue here, the coverage that you see is the standard off-the-shelf exome capture. And you can see a massive hole in the RPGR gene. This is the retinitis pigmentosa gene. And right in the middle where that arrow is, it's a known pathogenic variant. So the point here is just you have to know there's a huge gap there. You have to know what you're getting. And if you know what it is, you can fill it in. It's not as simple as just putting more capture probes on the capture chip, because some areas are caused by the GC chemistry that isn't changed by that. But if you know there's a hole there, you can pay attention, you can ask if it's covered, and you can know if you're getting the answer. If you can do it for one gene, you can do it for all the genes, and this is just an example to demonstrate that I haven't picked one in particular. The assumption has been, I think, that whole genome sequencing solves many of those problems, and indeed, overall, the coverage in whole genome sequencing is certainly more even, but as I showed earlier, it's still not perfect by any means for these 56 genes. It's a little better with the newer high-seq chemistry shown here. But in fact, when we looked at the X10, and I think maybe David, who's coming up after, may make some comment about this, but there's still some tweaking to be done clearly on the X10 chemistry because it's actually not as good as the high-seq chemistry from before for this metric. And so I think as we're continually reinventing these tools that have absolutely revolutionized what we're doing, there's a continual need for looking at and tweaking them to bring them up to clinical grade. This is an example just showing again these 56 genes. It's a little hard to see on the screen, so I maybe won't go into it in detail. But by paying attention to filling in the holes, you can see you can pretty much have 100% coverage at quality grade for all the 56 genes, whereas even whole genome sequencing struggles a little bit in order to cover those to the range that you would really want. So we also have to design better algorithms, and you've seen a great example of this earlier. This is from a simulated genome. So one of the answers to a gold standard is the NIST consensus approach that I mentioned earlier. Another is to just make a genome and put your own insertions and deletions in it, which is what my PhD student did here. You can see we're pretty good on sensitivity up at this range for small indels, but as you get beyond even 8 to 10 base pairs, the sensitivity drops off dramatically for the standard tools from the Broad Institute. There are good examples of people thinking creatively about how to solve that. This is one from the team you heard about earlier from here in Oxford. From Herton Lunter, Gil McVean, and others shown here. And you can see on the graph on the right, especially for deletions, but also for insertions, the Platypus tool is significantly better than the standard tools from the Broad. So again, an indication that we need to continue to tweak our tools in order to do better and get us to clinical grade. What we need to share on a global basis, the Global Alliance has been mentioned already. ClinGen is one of the NHGRI-funded initiatives to have a clinical repository of clinically relevant variants that we're part of at Stanford and many others too. And huge credit, I think, to Eric Green, who you heard from yesterday, for pioneering these various approaches. And in the last few minutes, I'd just like to tell you about one of the other new approaches that I've had the privilege of being involved in, and then one last other fun thing if we have time. The first is this, which is the Undiagnosed Diseases program that Eric and this gentleman, Bill Gall, were responsible for starting. And over the course of several years, it became very well known as a place that you could send the patients who really no one else could solve, the ones who'd been on an odyssey. And you could send them to the NIH and they'd be admitted for a week and have every test, including genome sequencing. And they published a paper. This is a little long in the tooth now, but you can see from the numbers here, new syndromes were defined, undescribed disorders were put together, new genes were found. Many individuals who'd spent hundreds of thousands of dollars and years of their lives without an answer found an answer through this program. And it was so successful that the Common Fund of the NIH made the decision to extend this to a network around the US. And I have the privilege now of chairing the steering committee of this with Bill Gall. And you can see the centers that are here. And we're just getting ready to open the gateway. The center can also take international patients. And we're actually attending an international session in Budapest this summer where we're looking at all the other countries. There's also, I think, one here in the UK to try and put these groups of people together. And putting them together, I think, is really important because if I have a patient in Stanford who has a particular group of symptoms, and someone else in Australia has a patient with a very similar group of symptoms, it would be very good if we knew about that, and we should be able to share not just that patient's details, but share the genetic information as well, so that we can look simultaneously for the same genes. And these sorts of programs that you see on the screen have come together to allow us to do this kind of global sharing. Putting the patient, and Eric mentioned this yesterday too, putting the patient in charge, giving the patient their data and enabling them to be at the center of their or their child's mystery disease is something that is really important to this group. So then just in the last 2 minutes I wanted to just tell you, again, Eric touched on this yesterday, part of the Precision Medicine Initiative, and if he was telling the story, you should ask him to tell you about this DNA model if you haven't already. He has a great story about it, but not for me to tell. But he mentioned that Apple had released ResearchKit. Actually, they made ResearchKit, which is a software development kit, publicly available yesterday, which for Apple is something quite unusual. It's open source, and you can go to the website and download it from yesterday. But over the course of a year, we were one of the academic groups. We worked on a cardiovascular app that was launched just a few weeks ago, and I want to show you a little bit of the data, the first time I think it's ever been shown in public from this app. And it was a long sort of secretive time because Apple, a very secretive company, we weren't able to tell anyone what we were doing. But one of the things that I was very happy and proud about was the fact that we've done consent on the phone. And the fact is, as shown here, are the different screens where you can on each screen find out more a little bit as you consent for the app. And by the time you get to the last screen, you're essentially signing an institutional review board application consent that then gets logged at Stanford University. But it's entirely implemented on the phone. You also have the Touch ID that is built into most of the iPhones, so we have the sort of ID power of that, so you don't need to continually enter a password. The study is based on questionnaires and then uses the sensors built at the low power sensor, the M7 and 8 sensor that's built into your phone to look at physical activity. We also do a self-administered 6-minute walk, which is shown on the other screen. One of the things about when Apple announces something is that people tend to pay attention. And this app has been downloaded almost 70,000 times in the last 3 weeks since it was launched. We have 35,000 consents, which, given that this study didn't exist, a month ago, is just a mind-blowing fact. And we didn't really have a team that was ready to deal with a study that has 35,000 people in it. But I've had the team knowing I was talking here, I've had the team working on some of the data. I want to just show you some basic stuff. Sanity checking, first of all. The physical activity goes up during the day. It goes down at night. It's good to know that that's the case. We can work out when people get up and when they go to bed. We also ask questions like, how happy are you? How satisfied with life are you? There's a map of the US. We mapped it to that. And just for those of you who don't know the states, I put them there. So not maybe surprising that California is pretty happy, and Florida, we get quite a lot of sunshine. Texas, pretty happy. The Northeast has had 100 inches of snow in the last... so they're maybe not quite so happy up here. Here's an interesting one. North and South Dakota. Look, North Dakota is really happy. And South Dakota, not so much. So I just Googled that from upstairs just a few minutes ago, and North Dakota turns out has oil. And they are really happy because they have lots of money, and South Dakota is not. I didn't know that. Being a poor Scotsman, I still don't really know my American geography in that detail. Okay, here's a final plug. One of the reasons that it's a joy to be here is we've been working over the last year or so with the group in Oxford around big data, Martin Landry and Rory and others and Peter. And spearheaded by the Dean Lloyd Minor at Stanford and John Bell here in Oxford. We ran this conference together last year. I actually was looking for the slide. Bill McVeigh is shown there. I was looking for the slide of Martin, but he hadn't apparently given in his biography, so that's why he didn't appear on the website. I don't know if he's in the audience, but this is the one. We have another one coming up from Stanford, and we're really eager to cement a strong relationship between the west coast of the States and Oxford in particular, but in the UK, and I have lots of motivation for doing that, being from here and this being home. I like any excuse to come back and be here. So, talking of that, thank you for having me. It's been a pleasure, and I'm happy to chat afterwards with anyone. So.